So in my group, we are developing new super-resolution microscopy technologies with the aim to investigate the structure and dynamics of proteins and their assemblies ultimately in the living cell. My name is Jonas Ries, and I'm professor for advanced microscopy and cellular dynamics at the University of Vienna, here at the Max Perutz labs, and we are here since January this year. We are an interdisciplinary team of physicists who build the microscopes, computer scientists who then write the analysis software, and finally biologists who use the technologies to investigate biological questions that only now become accessible because of the new technologies. Our biological research focus is endocytosis. That's a process by which cells take up molecules from the outside. Here, more than 50 different proteins in many copies self-assemble at the plasma membrane into a complex machinery, and then within seconds pull in the membrane and pinch off a vesicle. Up to now, it was very challenging to determine where all of these proteins are during this process. To investigate processes like this one, in my group we use a super-resolution technology called single-molecule localization microscopy. Here we label our target molecules with fluorophores that we can switch on and off, and then at a given time we switch on only a very small subset of these fluorophores, because this allows us to determine their precise positions, and from these positions to reconstruct a super-resolution image. However, this technique is rather slow, and therefore not too well suited at the moment to study living cells. To overcome this challenge, we therefore are following three new approaches. On one hand, we are pushing the speed of this technique, single molecule localization microscopy, so that we can use it for dynamic measurements. Then we are developing a new approach in which we take thousands of snapshots in fixed cells and then use AI, deep learning, to assemble all of these snapshots into a movie. Finally, we are using a new approach in single molecule localization microscopy that is called MINFlux, and that has a much better spatial and temporal resolution than our standard techniques. And this should allow us to directly detect movies at some point in the living cell. So in the past, we used MINFlux to investigate the motor protein kinesin and could for the first time directly image how it walks on microtubules in the living cells. We hope that ultimately our technologies will fulfill one of the main dreams of structural biology, and that is to directly observe how proteins undergo conformational changes during their action in the living cell. The technologies that we are developing are of course also applicable to many other biological processes. So as a group that focuses on technology and development, we therefore enable other biology groups to have breakthroughs in their research. And therefore it's very important for us to have many biological collaborations, but also to make all our developments readily accessible for other groups to use them.